I'm going to try to end with the story of fructose. But to begin, I'm going to pick up where I left off last time with what was I trying to feed you guys? Some sort of salmon, some sort of like seared salmon. Are you guys ready for the rest of that meal? So I had gotten your head in the game, and when I was talking about salmon, who likes salmon? Did I? Okay, so those of you whose hand went in the air, what was I doing to you when I was describing that lovely meal? Or maybe it's breakfast. I should talk about poached eggs and lox and bagels with cream cheese and arugula and chives and stuff like that, or whatever you find delicious.
We're doing that Pavlov thing.
That's right. Yeah, you were thinking about food. And that's the cephalic phase of gastric secretion. This is gastric secretion, gaster being your stomach, secretion being how your stomach makes the chemicals required for chemical digestion that happens in the stomach. So the first phase is cephalic. Cephalic means head, your head. The cephalic phase is getting your head in the game. And here we have this woman smelling a spoonful of her ragu or whatever she's eating there, and she's clearly enjoying that smell. So that whatever's happening up in her brain, in the like nucleus accumbens and the gustatory cortex and the olfactory cortex, etc., all that has this motor output that's coming down through the vagus nerve. Vagus meaning wanderer. It's the wandering nerve of the body, cranial nerve 10, because it goes to the heart, the lungs, the stomach, all over the viscera of the body. But on its way to the stomach, it stimulates a number of things, a number of cells into action in the stomach. So first of all, there's a host of cells, mucus cells, chief cells, parietal cells, G cells, all these cells in the wall of the stomach who each have a job of producing some sort of component of stomach juice. So mucus cells are going to make mucus, and mucus is a watery substance that has mucins, a complex saccharide, that are going to help lubricate and hydrate the food in our stomach. Chief cells are going to make an enzyme called pepsinogen. We're going to see what pepsinogen does in a minute. But the job of pepsinogen is it's basically a peptidase, which is an enzyme that breaks peptide bonds. And then the parietal cells make acid, hydrochloric acid. And then there are these things called G cells, which are going to produce a hormone called gastrin. Gastrin gets released into the bloodstream and is going to be an endocrine arc to this loop. That means a chemical signaling component of gastric secretion. The other half of the vagus nerve stimulation is physical. All right, so the first part here was activating a chemical digestion pathway, and the other is a physical digestion. It's going to start contracting the smooth muscles in the stomach and make the stomach move around like that. That's when your stomach is rumbling. The stomach is sort of squishing all the open air that is in your stomach, and you hear that growling sound, which is being elicited by the vagus nerve. All right. The next is the gastric phase, the part that actually happens in the stomach itself. So this is when you begin to eat. You're sticking food down there, and a number of things happen. First of all, the stomach starts to stretch, and you turn on these stretch receptors that it's like a positive feedback mechanism. It's going to get the stomach moving more and more. The rippling waves of smooth muscle contraction, autonomic involuntary muscle contraction in the stomach are going to get accelerated by the stretch receptors in the wall of the stomach. There are also going to be chemoreceptors, receptors that monitor how acidic the pH in the stomach is. And because those parietal cells are making hydrochloric acid, the pH is, it says elevated pH, and you know what? That's this slide. It's actually a lowered pH. The pH goes down, elevated acidity. I need to fix that. That was not my mistake, but I will fix it. Anyways, yeah, lowered pH triggers these chemoreceptors, and those stretch and chemoreceptors also feed back into the enteric or myenteric nervous system. That's the nervous system that lives in your gut. It lives in the walls of your stomach and your intestines. And it drives further production of the mucus, chief, and parietal and G cells. Then there is gastrin that's coming off the G cells. Gastrin is going to also be something, an endocrine hormone that's going to drive the production of pepsinogen and hydrochloric acid and will stimulate further waves in the autonomic smooth muscle fibers of the stomach wall. So you see both the cephalic phase has this long neural reflex from the brain. Then the gastric phase has this local neural reflex being driven by the myenteric plexus, and then there's an endocrine loop that's being driven by gastrin, which is being highlighted in green. All right, so this is as your stomach is filling up. The third is the intestinal phase of gastric secretion. This is essentially how we turn everything off. The stomach empties, and as the stomach empties, the presence of all these partially digested lipids, carbohydrates, and a lowered pH in the intestine is going to cause the production of a number of other hormones, downstream hormones, that enter the bloodstream and turn off the lights behind them, so to speak. These hormones, cholecystokinin, gastric inhibitory protein, or peptide, and secretin, these are all hormones that are going to turn off the chief cells, turn off the parietal cells, stop the mixing of the smooth muscle in the wall of the stomach. So that's the intestinal phase. It's how we stop gastric secretion. Questions on that? All right. Do you guys want me to tell the story of Barry Marshall? You guys know this story? Barry Marshall, just an interesting vignette about peptic ulcers. Back in the 70s, everybody thought peptic ulcers came because you were Kojak in your cop car eating a coney dog, and then you had to like throw the coney dog out the window and drive real fast, stressed out to the burglar or whatever, burning down the warehouse on the wharf. That's actually not how peptic ulcers work. They're not necessarily caused by eating some type of greasy, gross food or whatever. This guy here started taking tissue cultures from peptic ulcers and plating them out, and discovered that there was an acid-resistant bacteria, Helicobacter pylori, that was causing gastric ulcers or peptic ulcers. But nobody believed him. Nobody's heard this story? No. Nobody actually believed this guy. This was back in the 80s. And to prove his point, he tried to publish papers showing this, and it was just like such against the dogma about how food affected peptic ulcers that to prove his point, he cultured up a big Erlenmeyer flask of a big beaker of Helicobacter pylori, and yep, he did it. He just drank it down, gave himself a whipping case of peptic ulcers, and then took a course of antibiotics, and they completely cleared up. And for that, heroic, albeit kind of stupid, move on his part, he was awarded the Nobel Prize in 2005. So, yeah, all that happened back when he was like a graduate student, and that's the impulsive, go-get-'em spirit of a graduate student there. Certainly he would not have done that later in his career. But, yeah, peptic ulcers, you somehow come in contact with Helicobacter pylori, maybe through a coney dog that came in contact with it somehow. Who knows? Whatever food it was that caused you to be exposed to Helicobacter. But antibiotics solve the problem. All right, this slide, this is really just a filler slide so I can get from the stomach to the intestines. But the intestines, I want to be able to talk about the pancreas, which will lead me to talk about insulin, etc. Intestines have there's three parts: the duodenum, the jejunum, and the ileum. And what do I need you to know about them? Well, 90% of the nutrients that your body absorbs happens in the small intestine, not actually in the stomach. The stomach is where we break the components of our diet down into the nutrient pool. So we're breaking proteins into peptides and amino acids, and triglycerides are going to get emulsified, and carbohydrates are going to start to get acid hydrolyzed. But then all that moves into the intestines for further digestion and then eventual absorption. So because this is where all the absorption happens, the inside of your intestines have a really high surface area. And it's kind of fractal in nature. So if like macroscopically on the left, you see there are these ridges. They're called plica circularis. But then if you zoom in really close, there's all these little fingers called villi. That's a microscope image, a micrograph on the right, where you can see little food particles sitting down in the intestinal villi. All right, on to the pancreas. So the pancreas is important because it really is the driver of nutritional absorption and assimilation in the body, which is why, for example, pancreatic cancer is so disastrous for the people who have it. It's also an aggressive form of cancer. But the pancreas is really two organs in one. There are what are called endocrine and exocrine cells in the pancreas. Endocrine tissue is a tissue that's going to make some kind of hormone that is released into the blood, and an exocrine cell is a cell that's going to produce some sort of hormone or enzyme that gets released into a duct, not into the blood. It gets released into a duct. So first, the endocrine cells in the pancreas make insulin and glucagon. We'll talk about insulin very briefly when I talk about how sugar gets in the cells of our bodies. But for now, the exocrine cells are called the acinar cells, and they make up what's called pancreatic juice. And this juice gets released into the duodenum via this little duct that runs through the body of the pancreas. And it's made up of a host of different enzymes, alpha amylase, lipase, deoxyribonuclease, and these protease and peptidases. So those are pretty intimidating words, I realize that. But the four of them, those four different categories of enzymes, map onto the four categories of biomolecules that we talk about all semester. So amylase is simply an enzyme that breaks amylose down, which is starch. So that's how we break sugars down. Lipase is the enzyme, you may remember, enzyme that breaks, hydrolyzes the ester bond in triglycerides and releases free fatty acids. Deoxyribonuclease and deoxyribonuclease are not quite as important, but they break down RNA and DNA. And then the protease and peptidase breaks down proteins and peptides into amino acids. That's all that those are. So enzymes that break down all the nutrient components of our diet so that they can then be absorbed. Just to look a little bit at how this actually happens. So say we eat some starch. We eat a potato or a French fry, yeah, and that starch makes it into the duodenum, and the pancreas squirts out some pancreatic juice that has this alpha amylase in it. The starch is able to make it to the duodenum because the amylase that's in our saliva definitely breaks sugars down. There's salivary amylase, but it doesn't work at low pH. It doesn't work in the stomach. The acid turns that amylase off, and in fact, it actually digests the amylase itself. So by the time the starch gets to the stomach, it's been partially digested. Not a lot happens to it in the stomach, but then it makes it to the duodenum, and the pancreas finishes the job with its amylase, breaking that starch down into maltose. And a number of you probably tasted maltose in the lab, right? You guys remember that was the sort of brown syrup that was kind of sweet, kind of caramely in flavor. Yeah, maltose. Well, this is what happens in the duodenum. It gets broken down into maltose, which is just a disaccharide of glucose alpha-1,4 to another glucose molecule. And then there are these enzymes called brush border enzymes that are on the surface of the intestine. I showed you all those little villi, those finger projections in the intestine there. They have enzymes sticking off of them that are going to hydrolyze this alpha-1,4 linkage and just release pure glucose into the intestine, which will then get absorbed. Proteins use enzymes to break down. Our intestine uses these various enzymes to break down the proteins that we ingest. And the thing about them is they start as inactive proteins, right? These enzymes, and we call them zymogens. You can tell that an enzyme is an inactive zymogen when you look at the name and the end of it has O-gen on the end of it, O-gen, or sometimes the prefix pro. So trypsinogen and chemotrypsinogen are zymogens, and pro-carboxypeptidase is a proenzyme, meaning none of them are actually active. They need to be activated so that they can work on their substrates, on the proteins that need to be broken down. So trypsinogen gets hydrolyzed by enterokinase, which is another brush border enzyme in our intestines. It cleaves off this chunk of trypsinogen, which is like the regulator cap on it, and makes trypsin. And when trypsin gets released, that is the protease that is going to break down a bunch of other proteins. So trypsin can activate other trypsinogen, so that it has an autocatalytic activity. It can activate pro-carboxypeptidase and chemotrypsinogen to chemotrypsin and carboxypeptidase. And then all of those enzymes can work on the other nutritional proteins that are in the duodenum. Are there any questions? I know I'm blasting through this. Take a moment to take a breath here. Straightforward? All right. Cool. So here it is, the overview of the chemical events of digestion. This is sort of putting a lot of things. This is a pretty big overview slide. I just sort of talked about carbohydrates and proteins, how they get absorbed. We talked about lipids in the lipid chapter. But if moving from the top of the slide to the bottom, it's sort of like moving from our mouth through the GI tract into the bloodstream. So with carbohydrates, there's a salivary amylase that is going to break the starch down into smaller chunks that makes it into the small intestine, where pancreatic amylase takes over. There's going to be some other enzymes that'll break the maltose down into smaller monosaccharides. So lactase, we've talked about lactase before. Lactase is the enzyme that breaks lactose into galactose and glucose, and the absence of which is lactose intolerance. Maltase breaks maltose into the two glucose molecules. Sucrase breaks sucrose or table sugar into glucose and fructose. All those monosaccharides can get absorbed by the walls of your intestines and then just pass straight into the bloodstream. The lipids, I went through that in pretty great detail, so I won't belabor that again, but it's all right there. And then proteins, pepsin that is in the stomach. We talked about the production of pepsin just a bit ago, breaks proteins into polypeptides, which are then shuttled into the small intestine. Trypsinogen gets released by the pancreas, gets activated by enterokinase, and then it activates a lot of other zymogens and proenzymes, which break the nutritional proteins down into very small peptides and amino acids, which then can be absorbed by the walls of the stomach and are also able to pass directly into the bloodstream. The big difference here between carbohydrates and proteins versus lipids is that the lipids, to remind you, they don't go straight into the bloodstream because they're not water soluble. They need to be packed into these lipoproteins, the chylomicrons, which can then be absorbed into the lymphatic system and then eventually do pass into the blood. All right. So this brings us to the concept of the absorptive and postabsorptive states. So the absorptive state is this period in which your body is assimilating or absorbing nutrients, right? So, for example, now, I hope everybody had breakfast. You are in the absorptive state. Did anybody not have breakfast yet today? Okay, so you guys are in the post-absorptive state. Yeah, you're not actually in the absorptive state, and that means your hormone profile is a little bit different than everybody else who is sort of broken that fast for their day. But the absorptive state starts in the morning whenever you have breakfast. You take on a meal. Yep.
Does it have to be like full? Could I eat like one apple and now I'm absorptive?
Yeah, sure. You're absorbing the nutrients in that apple, and the pancreas needs to get turned on. It's going to release some. It's going to release insulin. Yeah, you're in the absorptive state. Post-absorptive state is dominated by glucagon. So, and absorptive state is dominated by insulin. So the absorptive state is when your body goes into anabolic processes, and I'll talk about what that means in a moment, but essentially the building up. And this is going to be one of the big themes, one of the big themes of the lecture here. But we use glucose for this whole period. The energy source in the absorptive state is glucose. The post-absorptive state happens when we have assimilated all of the nutrients that came from a meal, and catabolic processes, that means breaking things down, exceeds the building, or anabolic processes, the building up in the body. We use glycogen and fat in the post-absorptive state. So the energy that you are, whoever raised your hand, are in the post-absorptive state, you are burning glycogen if you still have any glycogen reserves in your liver from the night before, or a little bit of fat in the fat stores in the body. So usually after about 4 hours, a person leaves the post-absorptive state after a meal. So noon and 5 p.m. are typical times when you begin to get hungry. The body shifts the hormonal balance. And then at bedtime, you fall asleep and you've built up glycogen reserves in the liver to take you on your metabolic needs for the night as you rest. All right. So here is sort of an overview of how the metabolism in your body works. So catabolism is a process of taking larger molecules and breaking them down into smaller molecules. So taking proteins, breaking them down. Taking glycogen, breaking it down into glucose. Taking triglycerides, breaking them down into free fatty acids, and then chewing them up into acetyl-CoA to help produce ATP. Anabolism is building large molecules from smaller ones. So going from the nutrient pool, which is amino acids and monosaccharides and fatty acids, going up and building things like larger proteins, glycogen, etc. So anabolic processes are involved in like the maintenance and repair in your body, growth. If there's like some sort of secretion that needs to happen, or storing away reserves for times of post-absorption. That nutrient pool can also then be drawn down into energy production. And energy production is a catabolic process. It's a process whereby the mitochondria in your cells are going to break essentially carbon-carbon bonds, releasing that energy. And it's going to get stored and trapped in a molecule called ATP. I'll show you that in a bit. But ATP is the fundamental unit of energy, chemical energy, that the cells use for all kinds of things like locomotion and like all the little processes that the cell goes through, like endocytosis or exocytosis, or moving things around, shuttling stuff, reactions that are not energetically favored. And so that's an overview of metabolism. We're actually going to tease this this picture apart into two models. One model of metabolism for the post-absorptive state and one for the absorptive state. Yeah, but I want to zoom in for a moment on the mitochondria. So the mitochondria is a pretty interesting organelle in the cell. They believe there's strong evidence to suggest that the mitochondria was actually an independently living bacteria or prokaryote at one point in time that invaded eukaryotic cells and became a symbiote with eukaryotic cells. It produces energy, and in return, it gets to live it breaks small carbon fragments down and produces ATP, and in return, it gets safe haven in our cells. In fact, mitochondria, is everybody aware of that story? Is that like a redundant story from high school? Is that news to anybody? You're shaking your head. Well, anyways, mitochondria, the interesting thing about mitochondria for humans is that the mitochondria has its own set of DNA. The DNA that drives the production of proteins and whatever in mitochondria is not in your nucleus. It's not part of the 23 chromosomes. And what's even more interesting is that the mitochondria that you have all came from your mother. So mitochondrial DNA is only from your mother. The spermatozoa that fertilizes the egg during conception does not carry any mitochondria with them. All right. So they pretty much have the ATP they need to do a little swim and fertilize the egg, but they don't carry any mitochondria. So your ability to do this process of breaking down small carbon molecules and producing ATP, the like fundamental baseline of your metabolism, you can thank your mother for because mitochondrial DNA is matrilineal. Anyways, it's all about ATP, which is this molecule that you see right here. It's one of the nucleotide bases. You may recognize it from way earlier in the semester, except that instead of a single phosphate, it's got 3 on it. And it is this bond that I've indicated with the arrow right there that gets hydrolyzed when ATP goes from ATP to ADP and an inorganic phosphate. That's a pretty high energy bond that your body can utilize to drive any kind of reaction that needs a little oomph to get it to go. So the basic overview of how a mitochondria works is small carbon chains go in to the mitochondria, goes through this cycle called the tricarboxylic acid cycle or the Krebs cycle, and then it produces these reducing equivalents via what are called coenzymes, which I hope to talk about on Friday, what a coenzyme is, basically vitamins, which goes into this electron transport chain and produces, gives off ATP and water. One of the byproducts of the TCA cycle is carbon dioxide, right? So basically the mitochondria is oxidizing these small carbon chains with oxygen. It's not showing oxygen coming in, but that it should show an arrow with oxygen coming in because it's oxidizing carbon dioxide. So the carbon dioxide you breathe out with every breath it's the oxidized carbon from the fuel that your body burnt from the food that you ate. All right, so here's this combined map of catabolic and anabolic pathways. I'm going to tease it apart into absorptive and postabsorptive. But before I do that, I just want to give you the whole thing in one picture. So we've got this sort of nutrient pool here that's in the middle. These nutrients are absorbed by the cell. They can be amino acids, glucose, or fatty acids. And above them are the structural and storage and various functional components of the cell. And then below it is the mitochondria, which is going to be basically the energy powerhouse, the power plant of the cell, if you so to speak. So all the arrows in red are catabolic processes where we're taking something larger and breaking it down into something smaller. And the anabolic processes are in blue, where we're taking something small and building up a bigger structure from it. So in general, central metabolism is considered to be the carbohydrates right down the middle, the carbohydrate arm of this picture. So our modern diet, unless you are doing something like a ketogenic diet or some heavily protein-based diet, the modern diet is primarily driven by carbohydrates. It's a glycemic. We have a glycemic metabolism. And so understanding this middle arm that involves the carbohydrates is really important. It's the central dogma of metabolism. Going from glucose down to a small carbon chain, specifically pyruvic acid. So glucose is how many carbons? You guys remember?
6.
6, yeah, that's right. Pyruvic acid is 3. So essentially, we're just cutting glucose in half. The whole process, the catabolic process of going from glucose to pyruvic acid, is called glycolysis. I'm going to give you a bunch of words here that are going to be sound very similar to one another, but we're going to break them down so you can understand. Going from glucose to pyruvic acid is glycolysis. Glyco meaning sugar, lysis meaning cut. To lyse something is to cut it. Whereas you can go the opposite direction from, you can actually make glucose out of pyruvic acid, which is pretty convenient. And this is called gluco, meaning glucose, neo, meaning new, genesis, meaning creation. So you can make brand new glucose through gluconeogenesis, an anabolic process, from pyruvic acid. From glucose, you can go higher up and produce glycogen. Glycogen is animal starch. That process is called glycogenesis. I don't have that word on the slide, but breaking glycogen down into glucose is glycogenolysis. So, a lot of words that sound the same there. Important ones are glycolysis and gluconeogenesis, the opposite pathways, catabolic and anabolic, respectively. So then, pyruvic acid can shed a carbon dioxide in an energy evolving process. All of this, so this process of breaking glucose into pyruvic acid and then turning it into acetyl-CoA, this is what's part of what's called anaerobic metabolism. So say you are a sprinter, a power lifter, you are on a basketball court and you're sprinting down the court, or you are a soccer player and you're making a sprint, something like that. The energy that you're getting from that is coming from this part of this slide. So this part that happens outside of the mitochondria. You'll notice that none of it actually is calling for oxygen, right? There's no oxygen in any of those arrows, no oxygen pointing in. So anaerobic metabolism is metabolism that happens without needing oxygen. But the down, and that sounds pretty good, the downside of it is it's incredibly inefficient. It's only good for a very short period of time. It's kind of a wasteful way of extracting chemical energy from sugars. When the small carbon chain goes into the mitochondria, we invited them into our cells billions of years ago for a good reason. And that is because they're really efficient at doing this job. They are anaerobes, meaning they use oxygen to further oxidize the small carbon fragments that come in. So acetyl-CoA goes in, releases some carbon dioxide. We get these reducing equivalents that go into this thing called the electron transport chain. This seems like there's a lot of details here, but truly this thing called the TCA cycle, the electron transport chain, those are very complex chains of reactions that go around that we're not even going to look at all. But it is interesting, and you'll have to take my word for it without seeing the molecular details, that the electron transport chain requires some oxygen, evolves some hydrogen, but importantly, both the TCA cycle and the electron transport chain pump out loads of ATP. Okay, getting there. So here's the absorptive state. When we are in the absorptive state, we've had a meal, we've eaten some food, we are driving all of these blue arrows. Glucose is in our bloodstream, insulin is there, and the pancreas has released insulin, which enables us to absorb glucose directly from the blood. So we're going to break glucose down through glycolysis to release a bunch of ATP. That's all going to drive right into the mitochondria, and that ATP is going to help drive all of these anabolic processes. We're going to store some of this acetyl-CoA that's getting broken down from the glucose shunts back up, and we can build fats out of it. That's going to be important because if you have more calories than you need in terms of glucose, some of that, and you've stored all the glycogen that you can, some of those extra calories from the sugars you absorb get made into lipids through this pathway here, and that's enabled by insulin. So we convert carbohydrates into fats in the absorptive state. We can also build proteins out of pyruvate, the 3-carbon chain. And there's a host of enzymes that help drive that process. On the other hand is the post-absorptive state, and this is when we are taking things out of storage. We're breaking glycogen down into glucose for energy. We are taking pyruvate and making glucose out of it. If you are really glycogen deprived, you can break proteins down into amino acids, which can be made back into pyruvate and drive gluconeogenesis. We're not really doing the TCA cycle, the citric acid cycle, and the electron transport chain very much. We're not producing a huge amount of ATP. And the ATP we are producing, much of it is coming from lipid reserves. So we can break triglycerides down, fatty acids down into acetyl-CoA. So say you are in the ketogenic diet. You had talked about that. It was your thing. This is where we make ketone bodies. Say you're having a ketogenic metabolism. This is ketogenic metabolism as opposed to a glycemic metabolism, where we're getting the bulk of our ATP from the breakdown, the catabolism of lipids. Ketone bodies are other organic molecules. You see a few of them listed on the other side that can be taken up and utilized by cells in the body, specifically the brain, importantly the brain, instead of glucose. They are a carbon source that is derived from fat that can drive an alternate metabolism. And really this is an arm of our metabolism that comes out of the post-absorptive state. All right, I got there. Cool. Are there any questions on that? That was a little bit of science. I'm going to get less sciencey for a bit. Got 5 minutes to tell a story. No? Okay. So I want to talk about sugar again, and specifically I want to talk about fructose because glucose and sugars in general are so central to metabolism. You've seen now how sugar, if we have excess calories from sugar, that it can be shunted over through acetyl-CoA up into the production and storage of fats, right? So I wanna look at the obesity epidemic in the United States as it relates to sugar consumption. So this is a map I found showing global sugar supply in the form of sugar and sugar crops from the year 2007. It's a little bit kind of old data. But you can see the United States is really leading the way in terms of calories per person per day from sugar. So Americans have over 600 calories per day from sugar. That means the bulk of their, maybe a third to a quarter to a third of their calories every day are coming specifically from simple refined sugars. That's high, much higher than most of the rest of the world. Correlated with that is a rise in the body mass index of Americans. And so the body mass index is essentially a ratio of your height to your weight, and is a very rough estimate of body fat that you're carrying. And then you can see here that America, the mean body mass index for men and women globally is going up, but it's surpassed by the rise in the solid lines, which are men and women in the United States. And the problem is actually worse for men than it is for women in the United States. So here is a GIF just showing the geographic distribution of this rise. So the warmer colors are heavier body mass indices, whereas the cooler colors are lower ones. We're going from like the early 90s up through present. And you can see that what the biggest problem in the United States really starts in the southern and central United States, in the mid, spreads up into the Midwest, but New England is not completely immune from that problem. Well, where's all the sweet stuff coming from? Soda pop. We're drinking it, folks. Yeah. Yes, Mountain Dew. So you can see here that it's really the significant amount of the percentage of the total added sugars in the American diet, upwards of 50% for people 12 years old to 50 years old, for basically all adults, all adult-aged Americans. That added sugar is in the form of a drink, right? And that's sugar that has been divorced from the fiber that's in there, and that sugar is predominantly in the form of fructose, not sucrose, not maltose, not glucose, high fructose corn syrup. Why is it this way? Well, Big Soda has a lot of money at stake. Big Soda has donated $38 million into lobbying efforts to defeat various soda taxes that have been proposed in various cities across the country. That's over twice the amount of money that the federal government was able to spend on the U.S. Department of Agriculture's nutrition programs, which are trying to promote healthy eating. So you can sort of see, I have the representatives of the stacked up Coke cans versus some carrots on the right-hand side there. There's a lot of money. There's a lot of money driving things to be the way they are. But before I make the USDA out to be the poor beleaguered victims in this, they're actually driving it too. It's our federal government and corn subsidies. So they're talking sort of out of both sides of their mouth. On one hand, there is this MyPlate thing. Has anyone seen MyPlate before? You often see that in schools. They have this picture of what your plate is supposed to be, which shows, fruits, grains, vegetables, and protein, and dairy. That's the relative amounts of those things that you should be eating. Whereas if you were to redesign a MyPlate based upon the relative contributions of agriculture subsidies that the USDA is actually supplying to farmers, the real MyPlate would look like a plate full of enormous corn fritters, 80% of the subsidies, a couple peas in the corner, a teeny tiny hamburger, a little shot glass of milk, and then an after-dinner cigarette and a large cotton napkin. Those are representative of the agriculture subsidies that the USDA provides to farmers in this country, while at the same time supposedly promoting healthy, balanced diets. I will finish this story. I will finish the story next time. And I'll see you on Friday.